Hello students, today we are going to learn about transcription. Firstly, we will learn about the definition of transcription. Transcription is copying of genetic information from one strand of DNA to RNA. What is transcription? Transcription is copying of genetic information from one strand of DNA to RNA. Copying of genetic information from one strand of DNA to RNA, that is called transcription. Now, under this heading, we get two questions. One is about transcriptional unit and the other is about process of transcription. So, we'll study both. First, we'll study about transcriptional unit, and in my other video, I'll be going to make you learn about process of transcription. So, here we'll study about transcriptional unit. Now, what is the transcriptional unit? Transcriptional unit is the segment of DNA which undergoes the process of transcription, that is formation of RNA from DNA. It is the transcriptional unit is the segment of DNA which undergoes transcription, that is which forms RNA from DNA. Now, in before describing the transcriptional unit or the parts of transcriptional unit, we first need to mention about the conventions that we follow. Conventions means the things we have decided that we will do in this way, that are called conventions. So we will first discuss about the conventions we follow while describing the transcriptional unit. Then we will describe the parts of transcriptional unit. We will need to mention the conventions. What are the conventions? That the two strands, since a segment of DNA is transcripted and only one strand of the two things you need to remember. Only a segment of DNA is transcripted. During transcription, only segment of DNA is transcripted. That means as compared to replication, if we see, in replication the whole of the DNA gets duplicated. But here during transcription, only a segment of DNA is transcripted. Secondly, only one strand of DNA is copied, not the two. You may get this question separately, that why only one strand of DNA is copied and not the two? Because if both the strands of DNA get copied, then since the RNA formed from both these strands are going to be complementary, so they'll form a double-stranded RNA. And when they will form a double-stranded RNA, they'll be spliced off by the cells, because double-stranded RNA is like something different for the cell. And it will not transcribe that RNAs. So it will splice off that RNA. Secondly, if by chance both strands of RNA get translated also, they'll form different proteins, because both strands of RNA will be complementary to each other and the sequence of nucleotides present in them will be different. So they'll form different proteins. One will be the main protein that is required by the cell, and other protein will be a non-functional protein that may cause an interruption in the functioning of the main protein. So that is the reason. Number one, that if the two strands get copied, they'll become complementary to each other and will be spliced off by the cell. Secondly, if the two strands by chance get translated, they'll form two different proteins, because the two strands of RNA have different nucleotide sequence. They'll form different proteins. One the main protein, and one the functional. One will be the functional protein, and the other will be the non-functional protein. So these are the reasons. So that is a separate question you may get. Now we come back to transcription. So transcription, I described that is the process of copying of genetic information from DNA to RNA. Now we need to mention here that during transcription, only a segment of DNA is involved, and also only one strand of DNA is involved. So now while describing the conventions, we need to mention here that the strand of DNA which is going to be copied, which is going to form RNA, will be called as template strand. We'll call it template strand. Now, what will be the polarity of the strand? We need to mention here what will be the polarity. So you'll mention here that DNA-dependent RNA polymerase, which helps in forming the RNA from DNA, the enzyme that is called DNA-dependent RNA polymerase. DNA-dependent RNA polymerase can form RNA only in the 5'3' direction, same as DNA polymerase that we studied in replication. And also this is called DNA dependent RNA polymerase, like DNA dependent DNA polymerase, because it cannot synthesize RNA on its own. It needs a DNA template to form RNA, to form a sequence of polymerized RNA on it. So DNA dependent RNA polymerase. Now we need to write here the DNA dependent RNA polymerase, since it can synthesize RNA only in 5'3' direction. So the two strands of DNA out of which the one which has got polarity 3'5' will be forming RNA from DNA. The other strand will not be used in transcription. This strand which has the polarity 3' to 5' will be called as template strand, or we call it antisense strand, or we call it non-coding strand. The other strand, by convention, we call it coding strand or sense strand. Now you may get this question that why it is called coding strand, the other strand, since it is not going to, this is not coding for the formation of RNA, since still it is called coding strand. Why it is called coding strand? Because once the template strand gets copied and the RNA is formed from it, this RNA will have the same sequence of nucleotides as that of coding strand, because this RNA is the copy of is complementary to this template strand, and the other DNA strand is also complementary to this DNA strand. So both of them, the RNA and the DNA, will have same sequence. Other than that RNA will have U and instead of T, because DNA has thymine, and here we will have got uracil instead of thymine. So that is the difference. So this convention we need to write here, that the strand which has got polarity 3' to 5' will be called as template strand or antisense strand, or we call it non-coding strand, and the other strand that has got the polarity 5' to 3' will be called as coding strand or sense strand. Now one more thing we need to mention here, that whatever things we'll describe in transcriptional unit, we will take coding strand as a reference. Remember that the parts of, when we describe the parts of transcriptional unit, we will take coding strand as reference. Coding strand has the polarity 5', 3'. So we will take it as a reference. Now we come to the parts of transcriptional unit. Parts of transcriptional unit are, there are three parts: promoter, terminator and structural gene. So we will describe all of them by taking reference of coding strand. Now first we will write about promoter. What is promoter? Promoter is a region of transcriptional unit which is present at the beginning. First thing, it is present at the beginning at 5' end of the at 5' end of the coding strand, or 5' end of the transcriptional unit. We write here where, because we are taking coding strand as a reference. So where is the promoter present? It is present, we write promoter is present at the beginning of transcriptional unit at the 5' end. Understood? Then we will write what is promoter. Promoter is the region of transcriptional unit where the RNA polymerase binds along with the initiation factors. Initiation factors of transcription bind here, and we will study about the initiation factor in the process of transcription. So initiation factors bind here, and secondly RNA polymerase binds here. So we say this promoter is the part of transcriptional unit which is present at the beginning, and we call it upstream. What we call it? It is present at the upstream, at the beginning of the transcriptional unit and at the 5' end, at the 5' end of the transcriptional unit. Now we come to the terminator. What is terminator? It is at the end of it is present at the end of the transcriptional unit. It marks the end of transcription. We write here, it marks the end of transcription and is present at 3' end of the transcriptional unit. It is the region of transcriptional unit where the termination factors bind and where the RNA polymerase is released from transcription unit after transcription. And in between, there are structural genes that are going to form the RNA. Now the structural genes or the polypeptide. Now describing the structural gene will describe two things. One is cistron, about the cistron and the split. Now what is a cistron? It is the part of DNA which forms one polypeptide. So what is cistron? Part of DNA which form, or segment of DNA which forms one polypeptide is called cistron. Now the structural gene involved in the transcription unit of prokaryotes. We need to mention here structural genes involved in transcriptional unit of prokaryotes are polycistronic. That means from one transcriptional unit, whatever RNA will be formed, that RNA will code for more than one polynucleotide. And in eukaryotes, structural genes of the transcription unit of eukaryotes, they code for only one polynucleotide, so that is called monocistronic. Okay, so we write structural gene involved in transcriptional unit of prokaryotes. They code for more than one polynucleotide, so they are called polycistronic. You may get this definition separately also. What is a cistron? What do you mean by polycistronic and monocistronic genes? So in eukaryotes, the structural genes involved in the transcriptional unit, they are going to form only one polynucleotide, so they are called monocistronic. Second thing about structural gene we need to mention here, that in eukaryotes the genes are split. That means the express sequences of DNA are interrupted with non-express sequences of DNA, or the non-coding sequences of DNA. Thus, genes are split. You may get a separate question on this. The genes in eukaryotes are split. Justify. So, what does it mean? That the genes in eukaryotes, they are split. That means the express sequences of gene are interrupted with non-expressed sequences of genes. The express sequences of genes are called exons, and the non-coding sequences of genes are called introns. Introns do not appear in the processed RNA. Since when the RNA is formed from the transcriptional unit, it needs to be processed. We'll study in the process of during the process of transcription that it needs to be processed. That means these introns are removed from the process from the RNA, and then the final RNA is formed. That is called processed RNA. So in processed RNA, introns do not appear, but in processed RNA only exons appear. So we say structural genes of eukaryotes are split. That means there are some non-coding sequences between the coding sequences of gene. Coding sequences are called exons, and non-coding sequences are called introns. So that is all about the transcription unit, 3 regions: promoter, terminator, and structural gene. Thank you.